Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog. For Science for the Public, and tonight in our Public Science Lecture Series, which is sponsored by the Boston University College of Arts and Sciences this year, we have the pleasure of having Dr. Emery Brown present a wonderful lecture about states of unconsciousness. Dr. Emery Brown is the Warren M. Zapol Professor of Anesthesia at Harvard Medical School and Massachusetts General Hospital. And he's also Professor of Health Sciences and Technology and Professor of Computational Neuroscience at MIT. He has a full plate. He received his MD degree from Harvard Medical School and later a PhD in statistics at Harvard. Dr. Brown has received a long list of awards and honors, some of which we've listed on the Science for Public website for this lecture, but I'd like to mention just two that are very prestigious, and I think Dr. Brown may be the first person to be honored by both of them. The first is the 2011 Jerome Sack Award for Cross-Disciplinary Research from the National Institute of Statistics Science, and the second is the National Institutes of Health Directors Pioneer Award in 2007. Dr. Brown is also a fellow of three major organizations, the American Association for the Advancement of Science, the American Institution for Medical and Biological Engineering, and the American Statistical Association. What is important for us, however, tonight is that Dr. Brown has been a pioneer in interdisciplinary research and in three major fields: statistics, especially computational neuroscience, medicine, and bioengineering. This combining of multiple disciplines is very much the direction of science in the future, as you're probably aware. And Dr. Brown is widely recognized as a leader in establishing this kind of convergence. Dr. Brown's multidisciplinary approach is reflected in his research in the field of general anesthesia. At one level, this work is meant to benefit patients in the operating room, obviously, but that's just one level. Dr. Brown and his colleagues have made significant new insights about the brain, and particularly how the brain functions in different states of unconsciousness, specifically general anesthesia, coma, and normal sleep. The discoveries in this respect are important in brain science more generally and will have a far-reaching impact beyond that field. The general public does not have many opportunities to hear about this exciting area of science and its implications. So we are especially honored to have Dr. Emery Brown, one of the most distinguished researchers in this field, talk to us tonight, and it's a great pleasure to welcome him.
Oh, first of all, thank you so much for coming, Yvonne. Thanks very much for the introduction. I can't say I knew that I did all that, but anyway. So this is a real pleasure for me because I really like being an anesthesiologist. I mean it's a lot of fun. I mean I don't think people realize how much fun we have, and I don't think we even realize as anesthesiologists how cool what we do really is. I mean, in the course of a day, like I just came from the operating room. I was supervising residents in 2 rooms, each of which one did 2 cases, the other did 3 cases. So in 5 instances, we took people into a state of general anesthesia and brought them out. And we just sort of do it as a matter of course, go on, go home, have dinner, don't think about it. But when you really think about it, it's actually pretty wild. I want to give you some sense of that. I think that I hope in our field that we can generate more excitement around it, because it was a major contribution to medicine, the founding of general anesthesia, and now I think it has tremendous implications as a way of interfacing better with other areas of neuroscience. I'll mention this towards the end of my talk, because as we start to learn things about the brain through general anesthesia, they help us understand problems in other areas of clinical neuroscience, let's say psychiatry, neurology, neurosurgery. So that's going to be a theme that I'm going to go through today. So, but to make it very practical, I'm going to sort of go through a little series of vignettes here. Propofol's been in the news a lot for various reasons. And expert witnesses came to testify at the Michael Jackson case about propofol. But I want to actually tell you why it makes you unconscious. Why is it that when you give someone propofol, they actually lose consciousness, from the insights that we've gained from our research and also just from information that's out there in literature. Then I want to talk a little bit about general anesthesia and sleep. What is sleep? What is general anesthesia? Let's have a clear idea of that. And then towards the end, I want to talk about ketamine in particular, because ketamine is a drug of abuse which is used quite a bit. And we use it a lot as anesthesiologists as an anesthetic. And then I want to give you some insight into what the future might look like, what I call awakening, sort of reversing anesthesia. Right now, we don't reverse anesthesia. We let it wear off. And I want to show you what it might look like in the future. And then I'll just have a few conclusions. All right. So just to make sure I get the points across, I'm going to tell you what the punchlines are already. So why does propofol make you unconscious? Okay. So what I'm going to show you is that when you go into a state of general anesthesia, the brain goes into, let's say, general anesthesia or unconsciousness induced by propofol, the brain goes into a set of oscillations. And I'm going to talk about two of those, two principal oscillations. One is an oscillation that's at about 10 cycles per second, kind of like this. And that oscillation engages the thalamus and the cortex in such a way that it's difficult to transmit information, right? Then there's a second oscillation which comes about slow waves, they have a cycle which are about a little less than 1 cycle per second, a frequency of a little less than 1 cycle per second. And when they're present, it makes it difficult for parts of the brain to communicate. So now you've got two things which are preventing information from being transmitted. And these are empirical findings. I'm going to show you how we've come to realize that. And then the other thing I'm going to show you is that the other way so those are things which are happening up here in the cortex and then in the thalamus. And I'm going to show you down in the brainstem what happens is that it's very likely that the drugs are blocking the arousal pathways. So information can't go from the lower part of the brain to the upper part of the brain. So you have this multifactorial effect as to why someone's unconscious. And all the reason why, that if the drug is doing this, it's not a sleep drug at all. And you can see why, and this one turns out to be very, very potent, why it's something that you wouldn't want to take recreationally. Now I'll say a couple sentences about the recreational use of propofol. All right, so let me just begin with the definition. So what is general anesthesia? So we wrote this review last December for the New England Journal of Medicine. And we put forward this definition of general anesthesia because it's not necessarily widely accepted. And I think part of the reason we've had difficulty investigating our field is that there's not a consensus as to what it is. You kind of know it when you see it, but then trying to be specific about it. So this is a definition that we put forward. So you're unconscious, meaning you're not aware of what's going on. You're amnestic. You don't remember what happens to you. It's not supposed to hurt. It's nice if you're not moving around while the surgeons are operating. And then I think we often don't mention this last part, stability and control of various physiological systems. I mean, we keep you stable. Heart rate, blood pressure, stress levels, temperature. And because, I mean, just do a little Gedanken experiment. You could take the first 4 and they'd be synonymous with death, right? And like I like to say, people, you go to a cocktail party and someone says, oh, you're an anesthesiologist. You go, ha, 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 ha. You knock people out. You go, no, no. Any fool can knock you out. we bring you back. That's the main part, the stability and the control. And it's reversible, right? We bring you in, we bring you back. And what I want to show you right here now, just using a little bit of anatomy, a little bit of physiology, that the idea that we don't know how these drugs work just doesn't make sense. I mean, we're going to do a little basic exercise here. So how many people here have actually had general anesthesia? Actually had it? Oh, good. Well, show you this reflex that is called the doll's eyes. You turn the head one way, the eyes go the other way, and vice versa. Okay? So that means it sort of suggests very crudely that the nerves which control the eye movements are working. And the reason that's important is because what sits right next to them are the arousal centers, the centers that govern the sort of transmission of information from the brainstem up to the cortex and to the thalamus. All right? So, okay. Now I'm having just show you he's got normal strength. He's not weak or anything like that. All right? So if you've never seen this before, what we're doing now is, this is called denitrination or pre-oxygenation. We're having him fill his lungs with oxygen so that if something happens, it gives us a little extra time to go ahead to sort of do some emergency maneuvers if we have difficulty intubating him. So we'll do this for a little bit. To breathe for him now because he's unconscious. So let's just look and see how this looks right now. He wouldn't respond. He's not breathing. He's unconscious. And watch this. This is something that even catches by surprise the first time they see it, my anesthesiology colleagues. But in retrospect, they realize it. But watch his arm. His arm is like spaghetti. He just got propofol, all right? So three things happened. He lost consciousness, he stopped breathing, and all of a sudden he's flaccid. In fact, I was sitting next to a neurologist at a national meeting when I first got this video, and I stopped it before I showed this point, and I said, what do you think is going to happen to his arm? And the neurologist said, I think he's going to be like, spastic, right? Because that's what you normally think of, someone's kind of stiff or spastic in a coma. But this is actually a flaccid paralysis. So let's just look at why this is occurring. Let's just go and do a little anatomy, right? So these are the three things that we saw happen. He stopped breathing, he lost his muscle tone, and he became unconscious. So let's just dissect this a little bit by looking at the anatomy. All right, so these effects are happening here. So this is the base of the brain here, called the brain stem. All right, so starting right here, this area right in here is the midbrain. This little bulge out here is called the pons. The pons is a bridge between what was thought to be the top of the spinal cord and sort of the midbrain. Pons, like from Latin, like puente in Spanish, or pont in French. And then down here is the medulla. Okay, right down there. And then this area in here is the midbrain. And all these little colorful circles that I put here are the arousal centers. So these are the centers which transmit arousal information from the brainstem up to the cortex or to the thalamus. Okay, so we'll go over them in just a second. But let me just start here with the anatomy. So we injected the propofol in a vein, so it circulates through, finally makes it over to the left side of the heart, and then goes up the blood vessels to the brainstem. How did it get to the brainstem? Well, it got to the brainstem by this set of vessels right here. You see, this is a pretty interesting sort of set of vessels. This is the vertebral arteries. And this is one place where two arteries fuse to make one, the basilar artery. All right, so if you turn the brain over right here, turn it over, and you look right over here. Let's see if I can just get this. If you look right here, you turn it over. They're the two vertebral arteries. They fuse and make the basilar artery, and they're running on the underside of the brainstem. Okay, so one of the first places in the brain that gets the propofol are these lower centers. Just by the way the anatomy set up. Okay, gets there first. Now, so let's see who gets the first look at it. So right here in the lower part of the brainstem, these are the respiratory centers. The dorsal respiratory group and the ventral respiratory group. And so when the drug hits these sites, it can inactivate them, all right? This drug tends to enhance inhibition in the brain. So that's the first thing, right? Then, when you move a little further up, at this juncture here, this is the top of the pons, this is the midbrain. And what comes right out the sides there are the nerves which control the eye movements. So when neurologists, are looking at patients' eyes, it's not that they care so much about the eyes. They're trying to make inferences about the areas around the eyes. They're trying to make inferences about the other nuclei. And very often, they're trying to make inferences about these arousal centers here. And so the inference is, if something has damaged the movements of your eyes, then perhaps these arousal centers are inactivated or damaged also, right? And so that's what we're seeing here in this particular case. This gentleman's eyes are fixed in the midline, so the cranial nerves which control those eye movements, if they're affected, then by inference, probably these arousal centers are affected also, right? So that's kind of the logic. Now, the next part is a little, it's not as obvious, but there's very good clinical evidence for what I'm about to tell you. Sitting also in the brainstem here are relay nuclei, and these are two important ones. There's several, but they're relay motor nuclei. So information comes down from the motor cortex, goes down to these nuclei, and then goes down to the spinal cord. And there's a pair of them right here. These two up at the top here, the brown ones in this figure here. I can get control of my mouse. This is the Pontine reticular nucleus, and this is the pair. These are the medullary reticular nuclei. And what's interesting is NASA is interested in these pathways because you've probably seen the astronauts when they come back from being in anti-gravity for long periods of time. They're often slumped over. In fact, I don't think you ever see them give a press conference. They never hop out of the space shuttle and just, that was a great flight and all this sort of thing. It's always just a little while long before you see them because they've really lost control of those anti-gravity muscles. So they're the long muscles, ones that control like movements of the arms, like the cheerleader muscles, like, this sort of thing, this sort of movement, right? Okay, so what happens here? So these centers just sitting right here, so it also gets inactivated. So what's the clinical correlation for this? If you've ever seen someone, a patient, neurologists have patients, if they have a stroke, okay, and the stroke is due to a blood clot in like the basilar artery, right where it comes near the pons, when the person first presents, when they first show up, they're not stiff and rigid, they're actually flaccid, just like we saw in the picture there. So the point is that there's a lot of clinical evidence which sort of supports like what I'm telling you. So the idea that we can't understand how these drugs are working, where they're having their effect, just by doing a little analysis of just this little scenario, it doesn't really make sense that we should say that, right? So how else can we track the brain under anesthesia, right? So we put together this little sort of schema to show you what the EEG looks like because it was postulated back in 1937 that the EEG could be used to track the state of the brain under anesthesia. And it's only been in the last, let's say, 20 years or so that that's been used. But let me just remind you of a few things that you may have kind of seen in passing, but let's just get them firmly fixed in our heads because they'll be useful for the balance of the talk. This is someone who's just lying there, eyes open, kind of a nice sort of active EEG. Some people call it desynchronized, but that's probably not a good choice of terms, right? Because it probably is fairly coordinated, right? If you look over here to the right, this is someone, these are both awake patterns, someone who's awake, but now you see this very regular oscillation like this at 10 cycles per second. Now the way you get that is, this is called an alpha rhythm. And it's a rhythm that typically appears in the back of the head. If you place the EEG electrodes here in the back of the head, close your eyes, you'll get this very, very strong alpha rhythm. It's been known for years. The origins of it are not entirely clear. There's some theses out there about why it happens. But for our purposes, let's just say we know that it exists, okay? So that's you awake, either eyes closed or eyes open. Okay, now, let's go down the left pathway here. So let's say I give you a little bit of an anesthetic, okay? And what's ironic about getting a little bit of anesthetic is you don't start going into a state of general anesthesia right away. A little bit of anesthesia actually causes excitation, what we call paradoxical excitation. Now, when you first think of it, that sounds, well, that is paradoxical, right? But why do you go to a cocktail party? You know, why do people get high? How was ether actually discovered? People were having ether follies. They were using the drugs to get a buzz. I mean, this is actually kind of the EEG instantiation of the buzz or the high, okay? And there are various levels of being sort of discoordinate or what have you. And what you're trying to do is you're trying to get here, this is the cocktail party, you know, this is, you know, an overdose. I mean, that happens with alcohol, right? So that's not surprising when you put it in context. I mean, we actually know this. We have intuition about this. Now, once you increase the dose of the drug, the EEG starts to slow. And these are from an actual patient. I mean, this one isn't. We just drew this in for effect. But these are from an actual patient, one of our not patient, but a study subject, showing you the various stages of anesthesia. And what I'm going to be talking most about is this pattern here, where there's a combination of high-frequency oscillations like this and this slow oscillation. So that 10 hertz oscillation and this slow oscillation. And I'm going to contrast that with sleep in a little bit. But what I want to point out is just make a very, very specific statement about what's known about propofol. So it's been known for years that what propofol does is it goes into the brain and it helps the inhibitory interneurons. It helps the neurons whose purpose it is to decrease activity in circuits. And it does so by working at receptors that use GABA-A as the neurotransmitter. So when propofol is around, GABA-A is even more effective in inducing inhibition. And as you've seen, I've drawn this all over the brain. It's everywhere. It's in the brain, the spinal cord. So when the drug is given, the effects happen everywhere. Okay? And the differential aspects of the effects really depend upon sort of the local wiring, okay? Because all the brain and the spinal cord is not wired up the same. Okay, so that's what's happening here with propofol. We're going to go into this in some more detail. Now, I want to point out this brain state here. This is burst suppression. This is a very, very deep state of anesthesia. And in fact, with that bolus dose that we gave the gentleman in the little video there, he most likely went right to that state, as you saw in about 45 seconds or so. This is a state that you see in patients who've had a diffuse anoxic brain injury. You see it in hypothermia when it's induced, for example, either after someone has had a heart attack and they induce hypothermia to try to protect the brain, or when patients are on bypass, for bypass surgery, and they cool the patient down. If you look at the brain waves, it does this. It goes into this state where it's and then the bursts eventually disappear, and you have a silent EEG, and that's the quote-unquote isoelectric state. Okay? Now, one of the main differences between sleep, which is over here, and general anesthesia, is that you never see like burst suppression. So I'm going to talk a little bit more about these sort of patterns later on when I speak specifically about general anesthesia and sleep. But I put this here on purpose to, if we remember nothing else, just remember burst suppression you never see in sleep. I mean, you'll see things that look like the states here in anesthesia, but you'll never see burst suppression. Right? All right. So I want to go over some of the work that we've done to look at what happens to the brain under anesthesia. And this has been mostly done in collaboration with my colleague Patrick Purdon at Mass General Hospital there. So we set up a very simple study, conceptually very simple. Why not just image the brain in patients and look to see what turns off or turns on as they transition into loss of consciousness? And since we have the good fortune at Mass General to have one of the best imaging centers in the world, there's sort of no excuse not to do it. And so this is what we did. We gave a series of volunteers increasing doses of propofol, and you saw we gave that gentleman a bolus dose, and he lost consciousness within about 45 seconds. In this case, what we did was we protracted that out over a period of almost 2 hours, giving slowly increasing doses of propofol so that we can actually see the point at which the person lost consciousness. So that's straightforward, okay? Just slowly increase the dose. Where does the person lose consciousness? Now, the second part is you measure the EEG, because we have EEG patterns that we know are associated with loss of consciousness or changes in anesthetic state of the brain. And now you need a stimulus. You need something to tell you when the person has lost consciousness. So they're constantly listening to a tone and hitting a left button if it's a high tone and a right button if it's a low tone. So it's bong, bing, and they're constantly responding. So when they stop responding, we have a clinical indicator of when they've lost consciousness, right? Now, the next part of this was, as you saw, when the gentleman lost consciousness, he stopped breathing. So we want to put study subjects in a scanner. We want to have them all wired up. We want to track their EEG. We want to be able to deliver the drug in a controlled manner. And we know they're going to stop breathing, right? So what do? So what we did was we got patients to volunteer, or excuse me, subjects to volunteer for the study who have tracheostomies. So these are people who had been operated on before because maybe they had laryngeal cancer, maybe they had a dysplastic growth, maybe trauma to the larynx, and it necessitated them having perhaps a laryngectomy. And as a result, they're left with a stoma here. So we could just place a breathing tube right there before they went off under anesthesia, hook them up to the breathing circuit, and if they stopped breathing, we could just assist them, right? So we'd have exactly the same standards that you'd have in the operating room. And the idea, the suggestion to use these patients was given to me by my then chairman, Warren Zapol, who is a very, very clever, very clever physiologist, because he realized that we were groping with how to solve this problem. And then the rest of the study is also conceptually simple. If I'm playing a tone, I'm playing tones, so I'm going to activate your auditory pathway. So then just simply look to see how the activity in the auditory pathway drops out as you lose consciousness. That's the study, okay? Just very conceptually straightforward, right? So here's kind of the punchline, right? So this is a composite of the brain images made up of the 7 subjects that we studied in this investigation. Here's the baseline. This is before they lost consciousness, and this is after they lost consciousness, and here they are at the deepest level of loss of consciousness. So this is an axial slice, so you take the brain, turn it over, you're looking at it from the bottom, you make a cut across it like that. And then here we have a coronal slice down at the bottom, the corresponding coronal slice here. So the subject hasn't received any drug, and you see these yellow areas here. These yellow areas represent the auditory cortex being quite active, okay? And that activity continues even when before they've lost consciousness. They're still processing information, okay? Now here it looks like there's no activity, right? It's not that there's no activity. What we did was we set a level, and we said we're only going to show you in every image the activity that's above a certain level, okay? So if we drop that threshold, you would see more activity there. Now, what's interesting though is now here the subjects are at a profound state of loss of consciousness. And if you look back over here, in this area here, when they were clearly conscious, this part of the brain, which involves here what we're showing is part of that brainstem area, and then it's not really shown very well here, but the thalamus is actually more active when the subject is profoundly unconscious than when they're awake, okay? Now, we know the areas are active here because the information to get from the ear up to the cortex had to go through the thalamus. So even though you don't see it lighting up here like crazy in the imaging, it's active because the subject is hearing the tone and they're answering the questions correctly. So this was one of the first observations that we made, which was, sort of surprising. Could I say I predicted this? No. I mean, I knew you could sort of inactivate different parts of the brain and have unconsciousness, but I wouldn't have predicted this. So the thalamus is actually more active than when you are awake, all right? So, and here's just an illustration of that specifically. Here's the thalamus. It's actually quite active, all right? So that was a key finding. All right? And the why of this, I mean, our hypothesis as to why this occurred is the following. I showed you this pathway going from the ear up to the auditory cortex here, okay? what we didn't realize, we didn't think about when we designed the study, is that there are feedback pathways which come from the auditory cortex and project back to the thalamus. They project down to the brainstem as well. And these are negative feedback pathways, okay? So what happens is normally information comes up and it kind of gets sort of downregulated, kind of being modulated, sort of roughly sort of shut down a little bit. Let's say it that way. So now what happens is the anesthetic, in this case propofol, has its effect up in the cortex, and what occurs is this feedback pathway, which goes down to the thalamus and down to the parts of the brainstem, is no longer active, and as a consequence, those areas aren't inhibited, so they become more active than they were when the person was awake. All right? And it turns out there's very good neuroanatomy that goes along with that. But that's the first take-home message, kind of the thalamus is more active. All right. So that was a very hard study to do. All right. And we wanted to know something about what happens when you lose consciousness and recover consciousness. So we repeated the study in 10 subjects, increasing the dose of propofol, and then decreasing it so we could see what happened when you lost consciousness and when you recovered consciousness. They executed a series of behavioral tasks the same way. But now what we did was, instead of imaging the patients, or the subjects, we actually measured high-density EEG, so 64 leads of EEG on the subjects. Okay? And that's what this color coding map, this color map is here to show. All right? So I'm going to explain something to you, then I'll show it to you in a video. Okay? So this is what's called the spectrum. So what we're looking at, this is the whole study going from about 2 hours. And this is the EEG spectrum measured, or spectrogram, measured from the back of the head. All right, so let's just see if this sort of makes sense. All right, remember I said if you put electrodes in the back of the head, you're going to see a 10 cycle per second rhythm. Okay, that's exactly what you see. The person is lying there. We always have our subjects start with their eyes closed. So they're lying there with this 10 cycle per second rhythm. They get a little bit of anesthesia. There's the paradoxical excitation. And then what happens is the 10 cycle per second rhythm drops out and just goes to a low frequency rhythm. So it goes from being like this in the back to just this. Okay? Then, when the person recovers consciousness, it comes back. All right? Now, here's the front. In the front, you really don't see a 10 cycle per second rhythm. It's not there. However, when the person loses consciousness, you see a very strong 10 cycle per second rhythm in the front along with the slow rhythm, okay? This transition from having the rhythm, this 10 cycle per second rhythm be in the back of the head to the front, and it actually looks like this. It goes sort of forward like that. It's called Anterior Ization. This was worked out first pointed out about 30-some years ago by a couple of anesthesiologists who were studying monkeys up in the Mayo Clinic, all right? So I'm just going to show you the modern version of it, all right? So look, we have all these electrodes on this patient's head. So here, each one of these is an electrode area. Here's the nose, here's the left ear over here, here's the right ear, and this is the back of the head, okay? So here, we're going to be recording. I'm going to show you these spectrograms. So look, the person is lying there it is a he. He's lying there with his eyes closed. So here is that 10 hertz rhythm in the back, right? Okay. There's nothing in the front. Okay, so I'm just going to play this. I'm going to show you the whole study for this particular subject, okay? Baseline's gotten no drug, okay? Now he's gotten the first dose of drug. Now watch what happens. Look how the rhythm starts to appear in the front of the head now, okay? Now he gets a little bit more. He's just at the third level. Look, he's at the third level. This is already very, very intense in the front of the head. And look, it's in the same pattern is kind of like all throughout, sort of from the center here going out to the front in that direction, all right? And he's still not at the deepest level yet, okay? Now watch, so here's level 5. So this is, not only is it very deep level and from an anesthetic standpoint, but this pattern is very, very strong here. And look, the 10 hertz rhythm that was back here in the back of the head has disappeared. This is Anterior Ization. Okay, now, if you decrease the drug, let the drug wear off, okay? it disappears, and it goes back to the back, okay? So this has been known for years. There's nothing new here. It's just a sort of a way to present it, just to illustrate, kind of illustrate the point, all right? So the key, all right, so look, the two rhythms are there. So 30 years ago, people knew the two rhythms were there. The question is, what are they doing? Are they coherent? Are they coupled together? Are they uncoupled? All right, so that's where our research has given some insight into what's going on, all right? So this is just looking at one subject, going through the whole study. So here are the increasing doses of the drug, increasing levels that we targeted using computer-controlled infusion, all right? Now, you can see that there's the 10 hertz rhythm. All these little marks here indicate the person is responding to the questions correctly. And right here they stop responding. There's the paradoxical excitation. There's the start of this very intense 10 hertz rhythm. There's the slower rhythm, the slow oscillation or delta oscillation. The person stopped breathing right there. So we're assisting the breathing. We increased the drug level to here, dropped it back, and then the breathing came back. Eventually, the person started responding again, and the pattern looks like the pattern that we saw before we started. Now, these are target drug levels here. So these are levels that we were trying to reach based on a computer-controlled infusion. I just want to just point out a little factoid to you. In the Michael Jackson trial, they talked about the actual propofol blood level that he had, okay? So you can see, here's where the person stopped responding. So in here, the person's unconscious. Okay, so the blood level that they reported, this is on the autopsy report, was around here, around 4. Okay? So you see where he was. Okay? And I can tell you that we've given the person, the subjects in this case, several hundred milligrams of propofol. Just making a factual statement. Okay? That's what it takes to get to that state. Okay? All right. So what did we then figure out? Okay? So here's kind of one of the main punchlines. This is what was known about propofol. It enhances all of this inhibition. From our imaging study, what we found out was that these deep structures in the brain are actually quite active. Okay? Now, from this study here that I'm showing you now, we actually learned that at the 10 hertz rhythm, this area here across the front was very, very coherent. In other words, this 10 hertz rhythm was actually strongly, the rhythm that you saw across the front of the head was in sync across the entire front of the head. It wasn't uncoupled. Whereas the slow oscillation rhythm, it turned out, was uncoupled. Okay, so you have two states existing at the same time, right? So we went about trying to figure out what could be producing the 10 hertz oscillation like this. And this was a paper which we just published last year, in which we did some mathematical modeling. This is work actually done with Nancy Coppell here at BU in the math department, and Shenung Ching, who's my postdoc who worked on this. And what Shenung's work was able to demonstrate is that given the architecture of the cortex and the thalamus, and the connections between them, it's very likely that this 10 hertz oscillation is an oscillation being generated by the thalamus in collaboration with the cortex. So what happens? So roughly speaking, this occurs because the thalamus has two modes. So just to remind you, the thalamus is like this way station, right? Everything comes through it, going up, going down. I mean, the real analogy is like in soccer, where you have like a great center midfielder. A good center midfielder, all the plays go through the center midfielder, helps with the defense, helps with the offense, pushes the ball forward. The thalamus is very much like that. And so if you get like a central component of a system like that engaged, and it's engaged in, let's say, doing one thing, you've shrunk it down to a single bandwidth. Imagine, we're having communication now. I'm communicating across a very broad bandwidth. So now I shrink this down to, let's say, one particular frequency, 10 hertz. Okay? So it's like, that's all you have. So the cortex and the thalamus are sort of like locked into that rhythm, right? And then the other part that we figured out from this was that when you looked here at the slow oscillations, the slow oscillations, which we believe were coming from the cortex, are not coupled at all. So you've got two things. The little bits of cortex are here. They can't communicate with each other. And when they're communicating with the thalamus, they're doing so in a fixed oscillation. So if you say, why are you unconscious? What would be the dynamics of your being unconscious under propofol? We think that that's the case. And this is just to illustrate that particular point. Right here, see, each one of these is a study subject. This is the entire study here, okay? This is the people executing the behavioral responses, okay? So they're getting them all correct. Then they start missing some, getting none. So here's where they become unconscious. Now they start getting them again as the drug dose has been reduced, and now they're responding again. And you can see, just by showing you these 6 different subjects, that people have different response patterns to the drug. This person here went out, came right back. Look, this person here drifted down. It took a while to really lose consciousness. Was out for a while and took a while to come back. Took almost until the very end of the study to come back. But if you draw a line right down here, this is where the highest drug level is. Okay, and what this yellow line represents here, what this yellow line in each of these locales represents is coherence. It's the measure of coupling at that frequency. And so the highest that this coupling could be, if you look over here on the side, is 1. In other words, things would be perfectly coupled. But you can see that it's around about 75. All right? So it's a very tight correlation among all the sites somewhere. And the question is where? So that's why the color coding is important. So here's the color coding that I showed you right there. Okay? So you notice how on the front it's yellow and green. And so what Alan Simonser, my postdoc who did these analyses, showed is that it's all the yellow pieces here. If you read up from the bottom, most of the correlation is made up of the yellow parts, meaning it's coming from the front of the head. So at 10 hertz, a 10 hertz rhythm has the front of the head engaged when you're in deep stages of anesthesia. That's the conclusion from that. So this is what it looks like, okay? Here's the way to see it. So this is you're awake, okay? So just look right here at this neuron, this particular neuron. It's getting input from an inhibitory interneuron, and it's getting all these inputs coming out from different parts of the, let's say like the brainstem, okay? So there's a balance between the pluses and the minuses, okay? Now, propofol comes along, and propofol shifts that balance towards inhibition. It not only does it here, okay, but we saw that it does it down here in the brainstem as well, okay? So that gives you this particular state, okay? All right? And as I said, it's acting on these GABA receptors there. So you know how we talk about people getting addicted to opioids quite a bit. Okay, so opioids, we use them, morphine, oxycodone, we use them to treat pain, but there's also the risk of addiction. So the same thing is true for propofol, and perhaps, what's transpired with Michael Jackson has brought some of this to light. And so just like the opioids can be very effective in treating pain, they can also become addictive because they actually have effects on what's called the brain's reward centers, all right? Turns out propofol has a similar effect. And if you look at it, in addition to binding to those GABA-A receptors and sort of shutting down the brain, here's one of the main reward pathways, which comes from the top of the midbrain and going up to the cortex and going to the amygdala and the hippocampus. Remember, center of emotion, memory, short-term memory, and the nucleus accumbens, which is very, very important in reward. And the neurotransmitter that's involved is dopamine. So it causes this release of dopamine. So it's very, very interesting because, we know this clinically. Very often, people who've had anesthesia with propofol will come to and they go, oh, I feel kind of good. That wasn't too bad. They talk about this euphoria. And depending upon how long the sedation was relative to, the period of the operation, they may perceive this more or less, okay? And in fact, in this one study that was done in 2002 that looked at this, they showed that there's a dose-dependent effect in the amount of dopamine that's released from this particular area. So as you increase the dose of propofol, you actually get more and more dopamine release. So the sensation where the patients would probably perceive it is that they had sort of a whopping dose of propofol over a very short period of time. So the dopamine has been released, and after a while they come to and go, wow, you know, that wasn't too bad. All right. So that's probably what's happening there with you know, with the euphoria that's associated with propofol. And so I just put this in here just to show you. So this is actually one of the subjects, one of our patients from the operating room, like right here, okay? You give this bolus dose of propofol, and then the person goes right into the burst suppression, and here's that alpha pattern, and this is actually that slow pattern. So we actually are able to sort of see these things like on the monitors now. So this is what I've just shown you, this idea that why are you unconscious, slow waves, these 10 hertz oscillations, and then this piece here, which, I mean, this is the piece in some sense I'm kind of the proudest of, because you could sort of deduce this just by looking clinically at what's happening to the patient and just thinking about the anatomy without doing any of the studies, that it's probably having this very profound effect on these brainstem areas, right? So let me just say a couple words about sleep and anesthesia, all right? So very often when we talk to our patients, we say, we're going to put you to sleep or have you go off to sleep. And I remember once when I was a resident, you know, one of my senior colleagues got very upset at one of my co-residents because, you know, he said, I'm going to put you to sleep. And he said, you know, we put dogs to sleep. We don't put people to sleep. You know, we anesthetize people, right? And, you know, and we say sleep to patients all the time because we're looking for this euphemism. I mean, you can't go up to somebody and say, oh, excuse me, Mr. Scott, I'm going to put you in a reversible coma, you know, for your surgery, right? I mean, it just wouldn't go over well. But one of the difficulties is when anesthesiologists are hard-pressed to really say, well, what's happening, there's difficulty. So I just wanted to show you just some very, just to highlight now these obvious differences between sleep and anesthesia. I think they're important to understand. So this is sleep, all right? So you're awake, and then when you fall off to sleep, you go to non-REM stage 1, stage 2, stage 3. Now, I actually took this from a book, Purvis's book on neuroscience, which came out in 2008. And actually in 2008, they got rid of stage 4 sleep, right? That was the same year they got rid of Pluto. So it was a bad year for things that were on the margin, right? If you were out on the periphery, right, you were toast, right? So they're probably out drinking somewhere now, you know, because of that. So there are only 3 stages of sleep now, all right? So you come down, you go through the 3 stages, all right? Here's the deepest stage. Then you go back up and you go into REM sleep. Then you do that again. And this is kind of drawn correctly in the sense that you have your deepest periods of sleep. So stage 3 is a slow wave sleep, is thought to be the deepest stage, and you have usually about 2, maybe 3 bouts of that during the night, and the rest of the time you oscillate just between the REM and then the upper 2 stages of slow wave sleep, and then you typically wake up from REM. So that's kind of the classic way to think about sleep. Now, the 2 stages are quite different. So when you're in non-REM sleep, non-rapid eye movement sleep, things tend to slow down, you know, the heart rate, respiration, okay, and, you know, muscle tone is usually less, okay? REM sleep is just the opposite. It's rapid eye movement sleep. It was originally called paradoxical sleep. Respiration and heart rate are actually quite irregular. And it's very interesting to sort of see. So now if we go back over here, so this is what this would look like on the EEG. And I put these like this, not because I was trying to illustrate the relationship between the way you cycle through sleep normally, but just to illustrate what they kind of look like. So this might be the closest thing that looks like, you know, being either paradoxical excitation or being here awake. So REM sleep is actually an active brain state, which I'm going to show you in just a second, whereas non-REM sleep tends to be less active. So here, slow wave sleep, these look like the same slow waves that we just saw over here in anesthesia, but they're not. They actually have different properties, all right? And so the point that I want you to take home with this, if you've never sort of thought about this, it's really interesting to look at how we think non-REM sleep starts. So you start with non-REM sleep here. In this area here in the front of the brain call the preoptic area, and this is highlighting the ventrolateral preoptic nucleus, but it probably involves more nuclei than that, probably maybe even going as far forward, parts of the rostral forebrain, and here into the medial pontine, sort of the, I'm sorry, the median preoptic nucleus here. But the key idea is the following. This area becomes active, and it shuts off, and look what it's pointing to. Look where the arrows are going. The arrows are going to all of these arousal centers, okay? We started there, all right? It becomes active, and it shuts down these arousal centers. So that gets you into non-REM sleep, okay? Now, what has to happen for you to get into REM sleep is the cortex has to become active. So the projections back here in the back, at the top of the midbrain, sitting in the back, which go up to the thalamus and then on up to the cortex, they release acetylcholine. It's an excitatory transmitter. They become active, right? They make the brain active. They make the spinal cord, or they paralyze the spinal cord, all right? So the same projections here, which actually activate the cortex over here, actually cause the spinal cord to, the alpha motor neurons in the spinal cord to actually shut down. So what do you have? You have active brain, and sort of paralyzed body, okay? So that's why you can dream during REM, right? But you can't act on it, okay? Because otherwise you'd be like running all over creation. But when do you actually sleepwalk? You sleepwalk during non-REM sleep. So you can actually dream during non-REM sleep as well. But there's supposedly a quality difference in the types of dreams. They're supposedly much more vivid, like during REM sleep or what have you, okay? But the point is that this is kind of natural paralysis, if you would. So the body is naturally paralyzed during this state. And here, the cortex is actually quite active. So the states are different. So just think of it. Sleep is this. Oscillation, right? That's not anesthesia. Because you could go in there and shake the person like this, and they're going to eventually come to. Anesthesia is quite different. So when people actually pin me down and they say, well, you know, what do you actually say to your patients? You know, you don't say sleep, you know, you don't say coma. I mean, I actually just describe to them what's going to happen. I say, you know, we're going to induce general anesthesia. That means you're going to be unconscious. You're not going to know what's happening to you. You're not going to remember it. And you're not going to feel any pain. And during that time, we're going to be keeping your heart rate and blood pressure and all quite stable. And you won't be moving because it'll make it easier for the surgeons to operate. I mean, that's what I actually tell them. I mean, actually, I give them the definition of general anesthesia. The single neuron here. So one kind of main concept I just want to get across is whether you're awake, whether you're asleep, whether you're anesthetized, the activity in the brain is always a balance between inhibition and excitation. It's kind of just that simple. It's complex, but that's kind of a two-sentence statement of what's going on. So what do? We have all this inhibition, which is there from the propofol, and we don't go in and block the propofol. We go in and activate the excitatory pathway. And if the level of activity in that can exceed the level of inhibition, in principle, you should come to. All right, and so that's what we think was happening there. All right. So this is what I've told you. So we've looked at two drugs. Propofol sort of slows things down, controlled. Ketamine, which is very fast oscillations. We've come to an agreement about what general anesthesia is, what sleep is. You can see that it's a reversible coma. It has to be a reversible coma, right? If you think about what you're going to do to someone if they're having surgery, right? It's a very traumatic undertaking. And you'd have to be in a state where you're essentially insensate to be able to tolerate it, certainly humanely and safely, right? And I've shown you this last point, which is that perhaps we may have new approaches to the way we bring you out of anesthesia, right? And so, this is to me the anesthesiologist's view of the world, right? General anesthesia is in the center and everything else is kind of around the periphery. It's kind of like the old New Yorker version, the modern day New Yorker version of anesthesia, looked at this way. So what I think is really, really neat about anesthesiology, what I said in the beginning is, we can simulate all these states. So for example, ketamine is used to simulate schizophrenia, right? It's studied like ad nauseum in neuroscience as a model for schizophrenia, brain development, okay? One of the things that you see when brain development goes awry is the pattern of burst suppression. the state that you see at deep states of anesthesia. Okay? Locked-in syndromes. All right? We actually have drug combinations which can actually create locked-in syndromes. The combination like fentanyl and droperidol, it turns out, can create a situation where the patient looks perfectly comfortable, stable, but is actually stressed out to the max. You can't perceive it because you've controlled their physiology. When the drug effects wear off, they say, that was the worst experience of my life. Right? Depression over here. All right? In the case of low doses of ketamine are actually being used to treat depression. Very small doses, 30 milligrams, given over 40-minute periods, can actually be effective in patients that have chronic depression, not just new onset depression, but chronic depression. I showed you some examples of drug addiction, and the ideas that we've been looking at to maybe help people recover from anesthesia, some of those are actually borrowed from coma recovery, because it's very common to put a patient in coma on a dopaminergic drug to try to help arouse the brain. All right? So, you know, anesthesiology is a clinical neuroscience. And I think that one of the things which has been unfortunate, to be fair, well, let me just say it this way. We had some very complex problems to solve first. We had to learn how to take care of the heart, how to take care of the kidneys, how to take care of the lungs, right? So now we're in a situation where we've kind of solved those problems. And what we can do now is contribute to this field of clinical neuroscience, you know, by trying to understand how what we do works, and then using that to make contributions to these other areas that I've sort of outlined here. All right. So I know I've gone a little bit long, so I appreciate your indulgence. So thank you very much.